Skeletal muscle is composed of a hierarchy of structures. To explain muscle from large to small, we begin with the large skeletal muscle. Skeletal muscle is surrounded by a connective tissue called epimysium and is composed of little bundles of fibers called fascicles. If we pull off one of these fascicles, we find it is composed of little bundles of multinucleated muscle fibers, which are really fused muscle cells called myofibers. Each fiber is wrapped in a connective tissue called perimysium. Each myofiber is also surrounded by a membrane called the sarcolemma, which acts as the muscle cell membrane and has within it a bundle of myofibrils. These myofibrils contain the functioning units of muscle, called myofilaments. The sarcolemma plays a crucial role in muscle contraction. Here you can see it penetrates the myofibril through tiny invaginations called T-tubules. These T-tubules create a network surrounding called sarcoplasmic reticulum. The myofibril and contains all of the necessary ingredients for muscle contraction, which we will get to in just a moment. The long cylindrical muscle cell, or myofiber, covered by endomysium and sarcolemma, contains sarcoplasm. Myofibrils, peripherally located mitochondria, transverse tubules, sarcoplasmic reticulum, and terminal cisterns. The myofilaments are composed of a thin and thick fibers that are networked together in a functional unit called the sarcomere. The sarcomere is composed of thick and thin filaments which are systematically fit together, much like two combs stuck together with the teeth intertwined. The sarcomere is divided into different functional regions. Here you see the thick and thin filaments networked together. The M line is the attachment for the thick fibers. The A band is the dark region containing a length of thick filament. The I band is a light region containing a length of thin filaments. The H band is the lighter region containing thick fibers within the A band that has no overlap with the thin fibers. And lastly, the Z line is the end point of each sarcomere, which extends from Z line to Z line. The thin filaments attach to the Z line, also known as the Z disc. The mechanism of the sliding filament begins with an action potential arriving at the synaptic knob. This action potential depolarizes the axon terminal of the neuron, causing the calcium channels to open and allow a flux of calcium ions to enter the synaptic end of the neuron, of the axon. The increased concentration of calcium causes the synaptic vesicles filled with acetylcholine to move down and fuse with the cell membrane, which releases acetylcholine into the synaptic cleft. The acetylcholine is picked up by the receptors on the motor end plate of the skeletal muscle, which in turn opens the sodium-potassium channels and allows the sodium ions to enter the muscle. The leftover acetylcholine is then consumed by the acetylcholinesterase. This is an enzyme that breaks down acetylcholine to allow for another muscle contraction. The increase in sodium ions in the muscle cell initiates an action potential on the sarcolemma. Potassium ions are released, causing a repolarization of the muscle. The sarcolemma, seen here, is a membrane that surrounds and penetrates the muscle cell. These penetrations are called T-tubules. The action potential causes the T-tubules to stimulate the release of calcium from the terminal cisternae of the sarcoplasmic reticulum, essentially a storage unit for calcium ions. The calcium is contained in the sarcoplasmic reticulum by a chemical called calsequestrin. Calcium floods over the thin filaments of the myofibril and bind to the troponin, which pulls tropomyosin off of the active site, exposing the actin. The mitochondria within the muscle cell provide the myosin head of the thick filament with energy by converting ATP to ADP. With the actin exposed, the high-energy myosin head attaches itself to the active sites on the thin filaments, forming a cross bridge. The myosin head pivots to the low-energy state releasing ADP and moving the thin filaments over the thick filaments in an action called the power stroke. Another ATP molecule then binds to the myosin head, causing it to break free from the active site on the actin. As the ATP is converted to ADP, the myosin head is re-energized and is ready to cause another power stroke if an active site is presented. Calcium is then pumped back into the sarcoplasmic reticulum and attached to the calsequestrin molecule, which allows tropomyosin to again cover the active site, resulting in the relaxation of the muscle.